All right, so last day we talked about the difference between our short-term and long-term biological effects of consuming food. So again, when we talk about long-term, our appetite is a very social aspect. What drives us to eat is our interactions with our environment. Again, things like plate size, things like music actually will affect our appetite. Hunger is our biological drive to eat. This is to do with, again, the neuropeptide Y family. So there's the three neuropeptides, neuropeptide Y, PPY, and I forget the third one off the top of my head, the acronym for it. Satiation is what drives us to stop eating. So we're consuming a meal, what makes us feel full? Again, things like distension of our stomach, certain hormones getting released. And satiety is our long-term drive to stop eating. Again, this is to do with the release of hormones we're going to talk about today called Ghrelin. So again, all of this is decided, or all of this comes from neural impulses, hormonal or the endocrine system, and our immune system. So our ability of our gut-brain axis to recognize where we are in meal consumption, so have we just consumed a meal, has the meal emptied from our stomach and is in our intestines, all of this is communicated through these three pathways. We've already talked about the neuropeptide Y family. Neuropeptide Y induces satiety, makes us feel full. It also regulates mood. So when you get hungry, why do you get angry? This is all because of these hormones. This also interacts with our gastric compartment. It increases gastric motility. We have PYY, which is secreted along the entire gut-brain axis, but it's mainly secreted in the intestinal compartment. The other thing that's really interesting with PYY is that the gut microflora can actually interact with these transmissions and can actually induce different effects with satiety. The last one is PP, which is secreted mainly from the pancreas postprandially, so after we've consumed our meal, and again, it regulates satiety. Now, depending on the type of food we consume, if it's a fiber, that fiber fills our gastric compartment very quickly. It's very slow to empty, so we have satiation for a long time. But after that meal empties, we very quickly get hungry again. And these are very complex relationships between the macronutrients. And if you want to get more information, a really great article, again, is on the central nervous system interaction with foods. Once the food reaches our site, we have a cephalic response. So remember, when we brought the bread out, your stomach acid started to get produced. You started to produce pepsinogen. Your gastric motility started to change. You started to have neurological impulses that said you're hungry and you want to consume food. So this is the cephalic response to food. Cephalic response can actually induce physiological changes to the gastric environment or the intestinal environment. Now, when we talk about food in the oral cavity, there's two primary events that are happening. The first is the reduction of particle size. So we're chewing, we're masticating, we're breaking that cracker up like we did in class. While that's happening, saliva is being released, it's absorbing into that matrix, it's allowing for it to be lubricated so you can form a bolus and then swallow it. The other major events that are happening in the mouth is the secretion of two enzymes. The first is salivary amylase. Remember, salivary amylase is only able to break down glycemic. When we talk about glycemic carbohydrates, they are digestible carbohydrates, meaning they have an alpha-1,4 glycosidic bond. We're going to talk about this bond a lot when we talk about carbohydrates. It only breaks down the amylose or maltopectin to maltose. We do not have the enzymes capable of reducing maltose to glucose in the oral cavity. We're going to talk about where that happens today. We also have lingual lipase, and then remember we talked about that really cool amino acid triad, or that protein triad, the three amino acids that allow that hydrogen to be deprotonated, allow the electron shifts, increasing the electrophilicity of that nucleophile. This then allows that nucleophile or that hydroxyl group to attack that carbonyl. Again, we'll talk about this again in the carbohydrate section. Once it attacks that carbonyl group, it allows that fatty acid to be cleaved off. Now remember, we talked about that triglyceride having to fit into that enzymatic site, or that to reach that catalytic triad. So because of the geometric configuration of the molecule and that enzymatic site, we never cleave off the SN2 position. So the SN2 position in triglycerides always remains, with the exception of one enzyme. Anyone remember the enzyme, the lipase? Where's it from that cleaves off the SN2 position? Breast milk, right. So in the SN2, maternal lipase is the only lipase found in breast milk that's capable of cleaving off all 3 fatty acids. So we absorb 2 fatty acids and the monoglyceride. So again, it does not produce it doesn't produce the actual, it doesn't cleave the SN2 position. Once that food enters the stomach, we have to have it relax, right? So that gastric compartment is a relatively small cavity. Once you've filled it with food, you have to ensure that you don't regurgitate. So your vagovagal reflex relaxes the stomach, allowing it to undergo distension. Now this is where we pick up from yesterday, or from Tuesday. So when we expose foods into an acid environment, a lot of things will begin to happen to that food. The first being the disintegration of materials. So if we talk about plant-based materials, things like the pectin holding cells together will begin to be solubilized. So what I wanted to do today is show you a remarkable food. And this is the message I sent out yesterday. So here we have milk. So what is in skim milk? What are the primary constituents of skim milk? Water is the biggest, followed by protein. Well, skim milk, so we don't have a lot of fat. So we're putting in some milk. Do I want a little bit more? Maybe a little bit more. Okay, so I'm going to start this, and I'm going to go live. So if you go to Twitter now, you're going to be able to watch what's happening. How is this low battery? I just charged it yesterday. Oh, I'm not going to. All right. All right, so we've got milk. Now, we have, let's take a look. So in 250 mils, we have 9 grams of protein. That's remarkable. When you think about a protein contains amino acids. Amino acids can either be hydrophobic or hydrophilic. So in an aqueous environment, it's really, is that not live streaming to Twitter yet? It is? Okay. So what's remarkable about it is, think about this for a second. The majority of amino acids are fairly hydrophobic. They don't like to exist in water. So imagine you mix oil and water together. What happens? Very quickly, it separates. In a protein, when you have those hydrophobic amino acids, if they're exposed to water, they try to minimize their contact with water. So when you have 9 grams of protein in 250 mils, how is that not coagulating? How is it remaining soluble? So that protein has to fold itself in such a way that the surface contains predominantly hydrophilic, water-loving amino acids, and the core of that protein contains mainly the hydrophobic amino acids, right? So again, if we have this long chain of amino acids, we'll say roughly half are hydrophobic, the other half are hydrophilic. The hydrophobic ones don't want to be exposed to water, so that protein folds itself in such a configuration that's going to minimize its contact of the hydrophobic amino acids with water. So that's the first thing that's really unique about the proteins in dairy. The other thing is, if we look in here and we look at the amount of calcium, it doesn't actually say the amount, but it actually has 30% of our daily calcium. Calcium is extremely insoluble in water. Milk holds far more calcium than is what is soluble. So why is calcium in such high concentrations in dairy products without it separating out? So this whole idea of a casein micelle, which we'll talk about on Thursday, this supramolecular structure of these proteins interact with calcium and allow that calcium to be packaged within the protein. That's really, really important when we're talking about trying to get bioaccessible calcium. So dairy products are far better at delivering calcium than if you take, let's say, a calcium supplement. Now, what's really interesting with proteins, when they hit an acidic environment, they begin to denature. All proteins denature in different acidities, in different pHs. So what we've got here today is we're going to look at what happens in the stomach. So here's our pH meter. So pH of milk is around 6.6 to 6.8. So we're getting 6.7. Everything's nice and fluid. We're going to turn on the stir, which is, which one? This one here. So now we're going to start stirring the milk. Now, when that milk hits your stomach, the pH of your gastrointestinal fluids is going to be buffered by that milk, right? So your stomach is going to start out at a pH at around 6.5. Once that food enters your stomach, your brain gets signaled based on the vagovagal reflex. You get distension. So all of a sudden, those chief cells and the parietal cells begin to secrete acid and begin to secrete pepsinogen. Remember, pepsinogen is the zymogen. The zymogen is the inactive form. It has to be inactive because if the active form is released, it's going to actually begin to digest your cells. Oh, my little thing broke off. That sucks. All right. So we are dropping pH in a second. So here we go. We've got hydrochloric acid, and we're slowly going to start adding it. I'm going to pull this out so I don't pour it on my wire. So we'll put a little bit of acid in. So we've dropped down to 5.5. Again, it's still remaining in solution, right? Nothing's really happened. So we're giving it a second. We're going to drop it a little bit more. Hopefully I don't go too far. There we go. We're going down to 4.7. Now, we should be hitting soon the isoelectric point of that protein. Now, we'll talk about the isoelectric point in a second. So the isoelectric point, if you remember from first year chemistry, is when that molecule carries no net charge. So we've got a carboxylic acid end on one end of the protein and an amino group on the other end. So depending on the pH, that protein can either be negatively charged, right, if we deprotonate that carboxylic acid group, or it can be positively charged if we protonate the amine group. So depending on that charge, we can have three different scenarios with most proteins. With most proteins, as long as they don't have a charged side group. So, man, I'm already steaming up my goggles. I'm going to put in a little bit more acid. Here we go. So we're going to leave it for a second. So we're down to about 3.6. It's hard to see, but if you look at the sides now, and I'll see if I can get in with the Twitter. Do you notice the sides of the beaker all up here have that granulated material on it? So what's beginning to happen? The protein is unfolding because the pH is dropping. We're near the isoelectric point. So the isoelectric point is when that carbonyl group has a negative charge and the amine group has a positive charge. So we've got a negative charge on one end of the molecule and a positive charge on the other end. So what happens? Well, every casein will have that. So what happens then is that my positive charge is going to come and it's going to interact with the negative charge of another casein, and the negative charge is going to interact with another positive charge. And we create this network, and we get coagulation of proteins in the stomach. So eventually, it's going to go from a liquid to a semi-solid. And I have to turn up the stirring because it's starting to gel a little bit. It's hard to see. I'm going to turn that off and let it connect again. And now I'm just going to drop it right down to, let's see. So now the pH of an adult stomach, about 1.5. I've gone a little bit low, but again, when I do this swirl, you can see the aggregates that are forming. So again, that actually begins to curdle and turn into a cheese-like material. That allows for gastric emptying to be slowed. Why is that important? Well, when you're digesting foods, you want that transit time to be a long time. So the digestive enzymes are able to fully interact with that food. So when you talk about solid food particles, that food material has to be disintegrated. We have to break up those small particles to allow the digestive enzymes access to that. You can kill the periscope, because I think it is showing the point. I've got to find my pointer, which is here. So the acid in the stomach is not only inactivating different microorganisms, pathogenic or probiotic. So one of the reasons, if you follow the scientific literature right now, there's this whole argument on whether or not things like yogurt and probiotics really have any nutritional benefit, because a lot of the probiotics that you consume orally gets destroyed in the gastric compartment. So there's this whole article that just got published in Scientific Reports saying that traditional probiotics may not serve much of a biological function. Where it becomes very, very important in probiotics is through fecal transplants, which is the new trend if you're up with that area of naturopathic medicine. A lot of repopulation after C. difficile infections through doing suppository-based probiotics. So basically taking someone else's fecal sample and transplanting it into you. All right, so we talked about the anterocontractive wave. So that's the wave that comes in the bottom half, the distal end of that stomach, and it moves downwards and mixes the food. Now, when that's doing that, it's pushing the food material against that pleuric sphincter, and that pleuric sphincter, which is about 1 centimeter wide, constricts and allows a certain dimension of food material to move from the gastric compartment to the intestinal compartment. Now, the rate at which that material empties is partially dependent on the viscosity or the solid-like nature of that material. So imagine you take a glass of water versus a glass of milk's not a great example. What's a really viscous fluid? A soup. Let's say, sorry, what? Maple syrup, a good one. So take two different viscosities. The higher viscosity fluid is going to take a longer time to migrate through that stomach. If you have solid material, so if I eat lettuce, it's going to take a longer time to pass through the GI tract. So what they find is about 1,000-fold increase in viscosity changes the gastric emptying rate by about 30%. So it's not a linear relationship, but that's really important. So if you're talking about trying to induce satiety, you want to consume more viscous foods because they're going to remain in the gastric compartment longer. It's going to take a longer time for those to empty. So your stomach is going to remain distended for a longer period of time, giving you that perception of fullness for longer. Solids typically take longer to empty than liquids. Makes sense, right? Again, you have that solid, you've got to disintegrate that material, so it's smaller, the particles are smaller than the pleuric sphincter, so it can move from the gastric compartment to the duodenum. But it's not that simple. So here we go. So here's a really interesting study. What they basically did is they created a meal. The meal contained water, olive oil, croutons, cooked vegetables, and cheese. Half of the group was given to it as a meal, so a nice plated thing with, some vegetables, some croutons, some oil that you're going to eat. The other person got it in a smoothie. So they blended up exact same components, so the exact same amount of fat, Exact same amount of carbohydrates, exact same amount of protein. So, if the rate of gastric emptying was solely based on composition, we would all expect then that the solid food, the SM group, would transit more slowly through, right? Because it's solid, there'd be particles in it, but it's the opposite. So it's not as simple as just saying, if it's a solid, it does this, if it's a liquid, it does this. So what else plays a role? So again, we can't just say it's the viscosity or the solid-like consistency of that material that determines gastric emptying. Why? So what happens? So that food material transfers from the gastric compartment. The gastric compartment becomes less distended. The food then moves into the duodenum, and once it's in the duodenum, all different sorts of pathways start to get triggered, which we'll talk about in a second. So, once that meal has left the gastric compartment, so half an hour after you consume that meal, your gastric compartment is empty, all of the food has moved from the gastric compartment to the small intestine. After a meal, when that stomach goes back to its original shape, it releases ghrelin. Ghrelin is what we call the hunger hormone, and this is a relatively new area of nutrition and food science, and ghrelin plays a very, very important role in starting that cycle over again. So our blood sugar drops, our neuropeptides are transmitted, we begin to feel hungry. We eat a meal, that meal distends our stomach, we get a release of additional PYY, PPY hormones. This causes us to feel full, we stop eating. Our stomach empties, ghrelin is released, starting that whole cycle over again. So ghrelin is released after the stomach is empty, starting to trigger satiety again. But there's some other really important things it does. It tells your heart to work harder. Now, why would your heart have to work harder? Because when you consume a meal, all of that material goes into the duodenum, ileum, and duodenum, and we have to absorb it. It goes from the gastric, or from the luminal phase, so within our intestines, and it goes in, typically, to the portal vein. And that portal vein takes it to the liver for first pass metabolism. Now, if you have a whole bunch of nutrients, you need a lot of blood flow to the gastric compartment to allow those nutrients to absorb. So if you need additional blood flow, you need an increased cardiac output, right? Makes sense. So ghrelin, when your stomach empties, says the food's no longer in here because you're not absorbing anything in the stomach. It's saying that the food now resides in the intestines. There's going to be glucose, there's going to be amino acids, there's going to be fatty acids ready to be absorbed. So we've got to get the cardiac output going so we can actually start to absorb those nutrients. We inhibit insulin, so we don't want to take up too much of the glucose. We want to break it down and allow it to enter the bloodstream. We've got to stimulate pancreatic enzymes, right? We've got to have the pancreatin. We've got to have bile produced. And it actually plays a role in gluconeogenesis. So ghrelin not only says, hey, we are going to start to get hungry again soon, but it also triggers the rest of the GI tract to prepare to break down and absorb those nutrients. So it's remarkable that your body has found ways to repurpose enzymes. So one purpose is to signal satiety, one purpose is to increase cardiac output, and all of these hormones act on all different points along the gut-brain axis. And it's very difficult to take out an element of this kind of picture and still talk about foods. So we can design foods that undergo gastric emptying slower. So this means it delays ghrelin, which means you're going to feel full longer. And does anyone know what food right now is really important for changing the way our gastric compartment empties? What is the new fad? Everyone should be consuming what type of fiber? Kale. Sorry? Kale? No, that's not a fiber. Well, all fibers are indigestible. Soluble fibers. Soluble fibers. Beta-glucan, right? Oats. Why they're so good for you is they induce satiety. You feel full longer if you eat a meal containing high soluble fibers. So there's profs at Guelph, Amanda Wright and Dr. Douglas Goff, who are experts in this area. So what they look at is, let's say you eat a bowl of Cheerios for breakfast. So Cheerios have high amounts of beta-glucan. Beta-glucan is a soluble fiber from oats. So if you eat a meal of Cheerios for breakfast, does that impact how much food you then consume at a later time? So what they do is they have people come in and they say, okay, you're going to have a bowl of cereal. You're going to come back in an hour or 2 hours, and you're going to eat as much pizza as you want. And they watch how much pizza you eat. So people that don't eat breakfast cereals, don't eat soluble fiber, tend to eat more in the subsequent meal. So food scientists are playing with this all the time now. We're trying to design foods that will reside in your gastric compartment for longer. Again, that makes you feel full for longer periods of time. So these nutrient bars, these protein bars, all of this is being thought of when you're designing them, because you want that feeling of satiety. You want that feeling of fullness. Everyone's seen Ensure drinks, the meal replacements. Again, they're designed based on their interactions with the gastric fluids. Yes? Both. They're the same thing. Yeah. The feeling of fullness isn't just talking about distension. It's that psychological drive to eat. If you feel full, you're going to have a reduced psychological drive to eat. So they're kind of one and the same. So how do they know that ghrelin is responsible for higher food consumption? So we already talked a little bit about how we do these studies with soluble fibers, but in this study they took ghrelin, and half the people they injected ghrelin into, the other half they didn't. They just injected a saline solution. And then they set them loose at a buffet. And they measured, when you left, they'd put your plate on a scale, and they would measure how much food you took. People that were injected with ghrelin took more food on average than people who were injected with saline solution. So it shows you that ghrelin stimulates appetite. You want more food, so you consume more food. So ghrelin plays an extremely important role in satiety. Now, ghrelin is half the picture. The other half the picture, so again, increased concentrations of ghrelin. Ghrelin is released when the stomach is empty. So ghrelin increases appetite. Now there's another set of hormones that have been recently discovered called leptin. Leptin is secreted by adipose tissue. So the fatter you are, or the more adipose tissue you have, the more leptin you produce. Leptin decreases your drive to eat. They're antagonistic to one another. Ghrelin increases your appetite, leptin decreases your appetite. And these two work antagonistically to each other. But what happens is if you become morbidly obese, so in my case, my BMI is above 30%, my leptin begins to do really weird things to satiety. So this is one of the reasons overweight people tend to continually overeat, is because you have hormones that are saying you need to consume more food. So it's this really complex balance between these hormones and these neurological impulses that are triggering satiety and that are kind of allowing you to be satiated. And it's far too complex to get into here, but if you're interested, there's a really great article on all of the interacting hormones, because we've talked about PYY, we've talked about ghrelin, we've talked about leptin. There's a whole nother group of species of hormones that interact along this pathway. And this pathway is becoming very, very important in Western diets because obesity is so prevalent. So with the high obese population, we have people that are overproducing things like leptin. So when we talk about gastric emptying, we mentioned that the rate of gastric emptying is dependent on two things so far. The first being the viscosity of the fluid. The second being the particle size, or the amount of size reduction that has to happen in that food. But in that study we just looked at, where the food is blended up, the liquid food emptied slower than the solid food. So it's not simply what's happening in the stomach. We also have to look what's happening in the small intestine. So as that food fills the stomach, it stretches, it releases hormones, triggering gastric juices, triggering pepsin. Your antral contractive waves begin to push that food against the pleurae sphincter. It begins to transit into the duodenum, and ileum. And when that begins to happen, we get another whole set of neural impulses, a whole other set of hormones released. So again, it's not just what's happening in the gastric compartment, but all of a sudden a new set of hormones is released in the intestine. So when that food leaves or begins to leave the gastric compartment and reach the small intestines, the intestinal compartment recognizes that food has entered. So it then triggers a cascade of hormone effects. So the food now is in the intestines. The intestines recognize it and secrete two main hormones. The first is cholecystokinin, and the second is secretin. Cholecystokinin is extremely important. So now the food is starting to empty into the intestines. Cholecystokinin is triggered and then goes and acts on the gastric. Oh, I could actually use my little lab coat. It's released from the intestinal compartment, moving back up to the gastric compartment and slowing gastric release. So again, it's not just the viscosity and the amount of solid particulate that's in that food, but it's also how these hormones are triggered. And we don't understand how they're triggered. We know that when the food goes into the ileum, the duodenum, and the duodenum, we get an increase in cytokinin. We don't know why. So we don't know what our body is recognizing in that food, but the first thing it does is it slows down gastric emptying. So our nutrient is recognized to be in our intestinal compartment. It says slow down the rest of gastric emptying, and that plays another feedback. So we've got our viscosity, our particle effect, but then we've got our nutrient recognition and our hormone release, this cholecystokinin, which then slows gastric emptying, and all of the hormones associated with meal, with food consumption, all seem to have an effect on appetite suppression. Again, it makes complete sense. The food's moved into your intestines. What do we have to begin to produce? We have to produce pancreatic enzymes. We've got to produce pancreatic amylase, pancreatic lipase, pancreatic protease to begin to break down that food. So again, the release of cholecystokinin triggers the release of those hormones that then secretes. And the other thing it does is it controls your gallbladder. So your gallbladder stores what? Anyone remember what your gallbladder stores? Bile. So when that cholecystokinin is released, it acts on triggers, causes contraction of the gallbladder, and bile is secreted into the small intestine. Again, this is important. We'll talk about it a little bit. This is important for lipid digestion. Secretin does the same thing. Slows down gastric emptying. It stimulates the pancreas to produce bicarbonate. So remember, the pH of your stomach is around 1, 1.5. pH of your intestines is about 6.5. So we've got to neutralize all that acid. So it releases sodium bicarbonate. Bicarbonate is a great, great buffer. If you don't remember what a buffer is, go into your first year chem books. And a buffer is something that buffers acidity. So the small intestine, if we look at a cross-section of the small intestine, it's this highly folded organ. So again, it allows for a lot of surface area, but it allows for a lot of expansion. And then if we zoom in, there's these villi. And these villi are little protrusions that increase the surface area of that gut, of the intestines. Why do we need a high surface area? Yes? Maximize the absorption. So you're increasing your cardiac output to increase your blood flow, but you also want to maximize the amount of area to allow those nutrients to be absorbed. So we've got these villi, and on the ends of these villi, we have even smaller projections, which are called microvilli. And these microvilli are made up of cells that secrete different enzymes that allow for different nutrients to be absorbed. Does anyone know of a disease state where the microvilli are impaired? Celiac disease. So in celiac disease, when you're exposed to gluten, it causes a change in the morphology of your villi. So if your microvilli collapse onto themselves and decreases the surface area, what's going to be the most common indicator of celiac disease? Poor intestinal absorption. So if anyone in here is a celiac, chances are you got identified because you had an iron deficiency or a B12 deficiency, because your body is not absorbing as efficiently as it would if it wasn't exposed to gluten. So you actually get changes in the morphology of the microvilli in true celiacs. Now there's a lot of debate right now about the whole cause of celiac disease, which we won't go into, but it's really, really interesting that a disease state, something exposed to as simple as a protein, changes the morphology of your intestinal tract, which changes the ability of those molecules and those micronutrients to be absorbed. So in the small intestine, we have digestive enzymes that are being secreted. Those digestive enzymes are going to break down our proteins, they're going to break down our lipids, they're going to break down our carbohydrates. We get acid neutralization. Again, the gastric compartment has a very low pH. We have to neutralize that. We do so by secreting sodium bicarbonate. We also have to be able to absorb the different breakdown products. All of this is happening in the duodenum and in the ileum. Now, with carbohydrates and with proteins, the enzyme and the substrate are both released in the aqueous phase, right? Proteins are water soluble, carbohydrates are water soluble, enzymes are water soluble. So those reactions happen very easily. But in the case of fat, that fat is in a different phase than our enzyme. Our enzyme is in the aqueous phase and the lipid remains in the oil phase. So we have to secrete bile, and bile is an excellent emulsifier. Emulsifiers reduce the interfacial tension between an oil phase and a liquid phase to increase the amount of surface area. So bile is extremely important for the emulsification of fats, and the emulsification of fats is important again, just like the microvilli. We need a large amount of surface area for that lipase to interact at. So when we talk about lingual lipase, maternal lipase, and gastric lipase, the interesting thing about those is they don't require bile to actually undergo a reaction. Pancreatic lipase requires the addition of bile. It will be inactive without bile and colipase, which we'll talk about in a little bit. Now, the ileum is the distal part of your small intestine. This is where most of the absorption happens. This is also where the ileal brake happens. And the ileal brake is this whole new area of trying to understand how foods influence satiety. So, if we have a high ileal brake, the ileal brake is all the hormones that are being released. The CCK, the PPY, Ghrelin, Leptin, all of that plays a role in determining how fast our food moves from our gastric phase to our ileal compartment. And this is all controlled by gastric motility, but also is controlled by the hormones and the neural impulses that are triggered by food. All right. So we've already talked about salivary amylase. Salivary amylase is capable of breaking down the alpha-1,4 glycosidic bond of digestible carbohydrates. There's no amylase produced in the stomach. Pancreatic amylase is able to break those polysaccharides down into dextrins and dextrins down into maltose. It cannot break down disaccharides, just like the salivary amylase. So the amylases are there to reduce the size of the polymers of carbohydrates that are digestible. This then produces high amounts of maltose and other disaccharides depending on the type of polysaccharide we have. So here we have amylose, we add our enzyme, and it's going to produce 3 different compounds mainly. It's going to produce dextrins, which are long chains but shorter than polymers, typically less than 10 carbohydrates, 10 monosaccharides. We get maltotriose, which is a 3-carbon, not a 3-carbon, a 3-sugar, it is working, a 3-sugar carbohydrate, and we get maltose, which is a 2-sugar carbohydrate. Now, if we look at a amylopectin, so what's the difference between amylose and a amylopectin? Amylose only has alpha-1,4 bonds, so it's linear chains. Amylopectin has branch points. So these are what are called alpha-1,6 bonds. We don't break alpha-1,6 bonds very good. So instead of now producing the different compounds that we saw in amylose, we see the formation of oleosaccharides, so again, those dextrins. We see the formation of maltose, but we see what are called limit dextrins. These can't be broken down any further by amylose. So this means that the limit dextrin doesn't absorb as efficiently as the amylose. So if you eat a starch that's high in amylopectin versus high in amylose, it changes the glycemic index. And the glycemic index is the rate in which that glucose enters your bloodstream. So by producing limit dextrins, we're decreasing the amount of available carbohydrate for absorption. So it changes the glycemic index of that food. Everyone knows what a glycemic index is. Anyone not familiar what a glycemic index is? Okay, we'll talk about it in a little bit anyhow. So now we have these disaccharides and these different oleosaccharides. The brush border enzymes are what facilitate the digestion of disaccharides. So the brush border enzymes are found at the surface of the microvilli. So we have this brush border, and this brush border, so again, here's our microvilli, these intrusions, and this pink area on the outside is the epithelial cells that are residing between the interface between the gastric compartment and the luminal phase, or just the empty cavity. So all absorption has to happen through this brush border, and this brush border is extremely important. The first thing that the brush border does is it prevents self-digestion. So that brush border is this thick mucus layer that protects the intestinal cell lining from self-digestion. So it's this very thick mucin-like layer. Now, at the surface of the brush border, we release three major enzymes. We produce the maltase reducing, the sucrase reducing, and the lactase reducing. So we have three primary disaccharides, right? We've got sucrose, maltose, and lactose. Those are broken down into their individual monosaccharides based on the brush border enzymes. So we get the amylases, which reduce the molecules down to dextrins, to limit dextrins in the case of branched, and down to maltose. The disaccharides, including maltose, which is a product of digestion of carbohydrates, are then further metabolized at the brush border or at the interface where your lumen is. So at that interface, you're producing enzymes, and those disaccharides are converted into monosaccharides. The monosaccharides are then able to be absorbed. Disaccharides are not able to be absorbed efficiently. So when we talk about absorption, it all happens through facilitated diffusion. In facilitated diffusion, we need sodium to channel that glucose through. So we have sodium going through. Does anyone remember how we get rid of excess sodium from cells? What do we need? ATP, that's right. We need ATP to kick that sodium back out of the cell. So glucose pairs up with sodium at the transporter, and the transporter is able to transport it from the lumen, or from the inside of the small intestine, to that cellular material. So now it's inside that cell. So this one transporter, you don't need to know the names of the transporters. One transporter can do both glucose and galactose, and we need a second transporter to actually do fructose. Now, when we talk about the glycemic index, I just want to quickly show this now. So it's been sitting for a few minutes. For people near the front, can you actually see the separation of the serum layer and the separation of the coagulated protein? So you're actually starting to see separation. So if we leave this longer, it'll eventually completely separate out, where most of the protein will just be coagulated like cheese at the bottom of this. So again, this happens every time you consume milk. Now, once that monosaccharide is within the cell, we transport it out the other way using another set of transporters, and it's picked up by the portal vein. The portal vein takes everything directly from the GI tract to the liver, and it metabolizes it. So when you talk about drugs that you're taking, pharmaceuticals, if your drug is water soluble, it undergoes first pass metabolism. So it goes from the duodenum, and ileum, it's absorbed through the epithelial cells, goes into the portal vein, straight to the liver for metabolism. and we get a reduction in the bioaccessibility of whatever we're talking about. It could be a drug, a chemotherapeutic, it could be a natural bioactive or a nutraceutical. Now, what we try to do in the food industry is we try to put it into the fat phase, the bioactive. Why do we do that? Because fat is picked up not by the portal vein, but is picked up by the lymphatic system. And what's interesting there is if it's picked up by the lymphatic system, it goes through systemic circulation, all your cells are exposed to it, and then it gets into the bloodstream and undergoes first pass metabolism. So you can actually bypass metabolism if you can get a bioactive in a fat phase versus a water phase. So we can change the bioaccessibility of poorly soluble micronutrients, things like ECGC found in green tea. So why green tea is good for us is a polyphenol. You put that in the oil phase and allow it to bypass first pass metabolism, you get about a 30% increase in bioaccessibility. So again, by knowing the interactions between the food and the digestive system, we can begin to tailor and engineer foods that have specific performance enhancing attributes, meaning we can increase the bioaccessibility of nutraceuticals that we're adding to foods when they're poorly bioaccessible. So things like resveratrol, why red wine is good for you. Catechins, why chocolate is good for you. It's all dependent on the presence or absence of fat. All right. So lipid digestion. Again, the reaction is very, very similar to what we saw in the gastric, in the oral lipase, but now we need bile. So again, when we talk about oil and water, they are insoluble in one another. You mix oil, vegetable oil with water, you're going to have two very distinct layers. In your aqueous layer, you're going to have your enzyme, and your substrate is in the oil layer. That means we're going to have very, very little interface for that reaction to take place at, right? How do we increase that? We increase it by emulsifying the fat. How do we emulsify the fat? We emulsify the fat by the incorporation of a highly surface active molecule, meaning it's highly amphiphilic, meaning it's got a polar end and a non-polar end. So polar means it's water soluble, non-polar means it's fat soluble. So if I've got fat over here and water over here, the hydrophilic end of bile will sit residing facing the aqueous phase, and you have the hydrophobic end of the molecule residing in the oil phase. This reduces that interfacial tension. This means that when your stomach is churning and mixing the foods, that interface between the oil and water is going to go up because we have a surfactant or an emulsifier that's naturally produced. So when the cholecystokinin is released in your intestinal compartments, it goes and it acts on the gallbladder, it causes the gallbladder to contract, and it releases about 50 mils of bile into the intestinal tract. Now you can also bypass that. So you can bypass the gallbladder and you can actually have bile being secreted directly from the liver, but typically it's through the gallbladder and the CCK mediated contraction. So what is bile? Bile is made mainly of cholesterol, and it's your primary source to get rid of cholesterol. So when you consume, or if you have high cholesterol and you want to try to naturally lower it, one of the ways of doing that is by eliminating bile out of your GI tract. So what typically happens is the bile goes in, emulsifies, the triglycerides are later absorbed, and the bile is picked back up and recycled. The cholesterol is reused. But if I can bind that bile to something that's not digestible, so an insoluble dietary fiber, that fiber and the cholesterol interact, the fiber and the bile interact, and that is going to be excreted. If I excrete bile, that means my body has to use more cholesterol and it has to synthesize more bile. Now the only difference between bile and cholesterol is the addition of two hydroxyl groups. So we have the addition of two hydroxyl groups. So now in bile we have, imagine this is bile here. So we've got that conjugated ring structure. We've got four hydroxyl groups. Those four hydroxyl groups all point down. So I've got this carbon-based ring structure with four hydroxyl groups pointing down. This is extremely polar, right? Those four hydroxyl groups, that's going to reside on the aqueous phase. That carbon ring structure is apolar or non-polar. It's going to reside on the fat phase. So you've got the hydroxyl groups extending down. We're going to talk about in a second the two different attached groups. And then we've got this nice carbon-based structure which interacts very nice with fat. It's also conjugated to either glycine, a very, very polar amino acid, or taurine, which is an aminosulfonic acid. Some people will call taurine a amino acid. I'm not sure on the exact classification. But again, we look at the polar groups, the hydroxyl groups, the carbonyl groups, that is either on glycine or taurine. That increases, again, the ability to emulsify that fat. And again, we want to emulsify that fat so we can increase the surface area between the water phase and the fat phase. We want that because the enzyme is located in the aqueous phase of that system. So we need surface area for that enzyme to interact with that fat phase. And once that happens, we go from our fat and our water with a very little interface to these small droplets that have huge amounts of interface, which facilitate then the lipase reaction to occur. It gives it interface for that enzyme to get to that interface. The bile's there. It allows then the lipase to come in with co-lipase and actually begin to digest that droplet. So we consume our fat. We get a little bit of oral lipase. Doesn't do a whole lot. It's fairly inactive. It gets a little bit of activity in the intestinal tract, but it's insignificant. Less than 10% of our digestion is lingual lipase. In preterm infants and in infants, lingual lipase is very important. In adults, it is not. Gastric lipase, again, it represents about 30% of our lipid digestion. So about the rest, 70%, is pancreatic lipase. Pancreatic lipase requires co-lipase and bile for it to be active. It will not access the interface without it. So once the bile, so our yellow droplet above that nature paper is our fat. Bile then goes, the bile again has the 4 hydroxyl groups, which are polar. They reside on the aqueous phase. That cholesterol ring, which is those aromatic carbons, those aromatic cyclic structures, they reside on the fat phase, reducing the interfacial tension. This then allows co-lipase, which prevents the inhibitory effects of bile on lipase, and lipase then interacts with that interface and begins to cleave off the 1 and the 3 position. Again, 2 monoglycerides are a byproduct of digestion. We do not break down that fatty acid at position 2. So we absorb 2 fatty acids and 1 monoglyceride for every molecule of triglyceride we consume. Now, so again, these different lipases have different activities to different chain length fatty acids. So again, we talked about that catalytic site. That molecule has to reside in that catalytic site. That's a stereometric or a morphology problem. So for that triglyceride to fit into that catalytic site, it's going to depend on the length of the fatty acids. So different lipases are specific to different chain length fatty acids. Cleave off position 1, cleave off position 2, we're left with the 2 monoglyceride. Both of these are absorbed, and within the cell, within our lining of our epithelial, they're reassimilated. So we have the enzymes capable of putting those 2 fatty acids back onto that glycerol. Those then form little pockets of fat. They then move through the cell, incorporated with some glycoproteins and some proteins, and we form the chylomicron. So HDL, LDL, VLDL, chylomicrons, right? You've heard of those from first year biology, right? VDL, very low density lipoprotein, HDL, high density lipoprotein. Your HDL-LDL ratio dictates your health, part of it, your cardiovascular health. You want a high HDL, you want a low LDL. You want really low chylomicrons. So chylomicrons then go to the liver to be oxidized later on. But these are picked up not by the portal vein, which we saw in the carbohydrates. These are picked up by the lymphatic system. Again, this is important because it bypasses first pass metabolism. So if you're incorporating a bioactive, you can bypass the liver by getting it to be taken up by the lymphatic system. This is a very, very active area of research right now. All right, we're going to stop here. This gives us right on time.